first and foremost, the work that the breeding program did in Haiti to develop those new varieties was so important to bring this food security crop back to Haiti. So part of what the paper does is just highlights the amazing work that the breeding program did. For my lab, as a geneticist, what we were able to do was kind of leapfrog off the success that the breeding program had, really with just traditional breeding methods done very, very well. We were able to go in and identify the signatures in the genome of sorghum for what those breeders selected for. And based on that, we were able to develop with the Haitian breeding program new technology that will help move the aphid resistance more quickly into their new varieties, to identify new sources of aphid resistance, which is an important concern of theirs, and also to help translate those discoveries to other countries, other breeding programs. What we do in the program is really try to design backwards from the needs of those users. Nobody knows the needs of the farmers, the consumers in these countries better than the breeding programs. And then we take what we're hearing from the breeding programs about what's working and what's not, and we try to find where some of the technology exists already in the US that solves potentially one of their problems. And if the technology doesn't exist, which in many cases it doesn't, we work hand in hand with the breeding programs in country to develop the technology that they need. Now, luckily, often the same technology that they need in the developing countries is the same technology as what's needed in the US sorghum industry. And so that's where we get to the win-wins like this situation. Sorghum is one of the most important crops in Haiti, along with corn and rice. It would be one of the staple cereals. An important thing about sorghum as well is that it is one of the homegrown crops in Haiti. In recent decades, there's been a lot of food imports to Haiti to deal with the food crises, but sorghum has been something grown by the Haitian people to feed themselves, and it's also the most drought tolerant of the staple cereal crops. Haiti, like other countries in the Caribbean, has some humid areas, but it's actually also got some very drought-prone areas. And a lot of the agricultural regions are drought-prone. That's why sorghum is such a key part of the food security of Haiti. In our paper, we showed how the sorghum production just dropped precipitously in just a couple years when the aphid arrived, almost disappeared. Let's take the example of the discovery of the aphid resistance gene, RMES1. That was actually done initially by a lab in China. They published that in 2013. But it was a really great example of the difficulty of transferring knowledge and discoveries. That discovery was made in a breeding line in China that's not available in the Americas. So you could read the paper and you could see, yes, that there is a gene involved in resistance at somewhere around here in the sorghum genome. But just because that exists in one line in China, it doesn't mean that you can actually do anything automatically with that knowledge in Haiti or in the US or anywhere else. But what we were able to do by basically layering an understanding of the history of breeding sorghum breeding over the last 50 years with some of these new genomic technologies, we were able to test the hypothesis that the same gene is involved in resistance in Haiti and the United States. Armed with that knowledge, then we were able to develop the markers and then confirm the idea that the same gene is actually involved in resistance from China to Haiti to the US and also back to the original origin of sorghum in Africa. different public breeding programs had different populations, right? They were kind of all on their own island. We were able to kind of trace where there was connections among the islands. And so then we knew, once we had done that, that the marker should work for many breeding programs across the world. Once we did the experiments to prove that, then we had a technology where we could say, yes, if you have certain breeding lines that you used in the history of your breeding program, then this marker should work for you.